My name is Dr. William Schwieterman. I'm the CEO of Stream Biomedical. One of the biggest public health challenges we all face in the world today is the absence of any effective therapeutic agents for the treatment of neurodegenerative disease. All of us, of course, know someone who's suffering from this, be it Alzheimer's disease, stroke, TBI, or other modalities. We at Stream are presenting today a novel solution to this problem. It's a biological solution to a very difficult problem using the body's own natural innate immunity repair mechanisms to restore homeostasis to the neurovascular unit. And what I'm going to be presenting to you today is new. It's novel. It's a first-in-class molecule that we ourselves have developed at Stream after in-licensing this product from Texas A&M, and it's proving to be a very potent, pleiotropic, and broad-spectrum protein that works across a range of preclinical studies. So, as I mentioned before, many of the difficulties in the past with moving forward with therapies for neurodegenerative diseases have failed because of the specificity, really the isolation of the target, the absence of pleiotropy, the absence of a broad spectrum effect across the NVU. This product of ours, LG3, is a solution to that particular problem. It's a recombinant biological protein derived from the matrikine family of molecules. Matrikine meaning extracellular matrix, kine comes from cytokine. These are proteins that are cleaved off at the first signs of injury from caspases and work really as hormonal drugs that cross the extracellular matrix to the inside of the lumen, repairing the tight junctions to the pericytes and restoring the epithelial covering of the blood-brain barrier there, the abluminal side, thereby restoring homeostasis to the NVU and thereby reducing inflammation and a variety of other factors. In a nutshell, this is a new class of molecules that's pleiotropic, it's potent, it's active very quickly, and it acts by a novel mechanism for which we have exclusive IP rights across the board. In this presentation, I don't have time to present all of the data on this, so I'm going to show you a sampling of different data that we have. Our plan going forward is actually to study this first in traumatic brain injury. Of course, a drug like this, a biological protein like this, and I worked at the FDA for 10 years. I was responsible for the approval of Enbrel, Humira, and other particular drugs. This is reminiscent of that kind of a paradigm-shifting drug going forward, is amenable to multiple different types of neurodegenerative disease, both acute and chronic. Our first study is planned for 2026 in normal healthy volunteers. We're ready for the clinic, but we quickly want to use this as a launching pad to other sorts of chronic neurodegenerative diseases, including CTE and Alzheimer's disease. Our exit is going to be after the readout of this TB trial at the end of 2027, where we can go on then to other modalities. Now, let me just walk you through a little bit about this particular protein. It repairs the blood-brain barrier, as I said, by restoring tight junctions and pericytes on the abluminal side. It protects and stabilizes brain cells. It's an anti-apoptotic agent. It blocks inflammatory cascades. We have initial data showing a diverse and new anti-inflammatory approach in the field of stroke, and it reactivates natural neurorepair. It works through integrin receptors, alpha-2 and alpha-5 beta-1 receptors on multiple different cell types within the neurovascular unit. And we've been working with a number of different collaborators around the country on this. We're now going out after having established IP with this particular class of molecules. I'm going to show you some of the results of some of the work of this. So we've published some of our initial work in a journal several years ago that shows the seminal findings of this particular class of molecules. So one of the interesting studies we did was out of Texas A&M. These are stroked brains from mice. On the left, you get a placebo sham control. On the right, you give LG3, which is this 21 kilodalton matrikine product. You see immediately that the vascular fluorescence represented in green here is hypodense with the sham control. There's a loss of vascular tissue in this particular model, versus LG3, given again shortly after the stroke, preserves, restores, repairs, preserves homeostasis to the neurovascular unit. We see this across a different range of a number of different animal studies. Again, I can't present all these data here in this presentation. This is a traumatic brain injury model. Similar findings as before. You preserve brain tissue, we restore function in these particular animals. If you see on the bottom here, I don't know how well it projects, but you prevent the ecchymoses associated with BBB simply because you've now sealed that blood-brain barrier as a primary mechanism of action. We see similar things in an Alzheimer's model. When we give this over a short period of time, we clear amyloid, particularly out of the hippocampus. Interestingly, consistent with the neurovascular hypothesis of Alzheimer's disease, we restore function, and we actually have new nerve cell generation in the caudate nucleus with this particular drug. Consistent, again, with a natural endogenous molecule that's in all of us. This molecule is upregulated in us. When we fall to the ground, we get hit in the head. We're using this recombinant protein in supraphysiologic doses to these particular animals, and that's what we want to do to patients in our clinical studies. Here's an example of the effect in stroke. So we have 80% protection of the infarct in these stroked animals, and they're also associated with increased function, data not shown here. On the bottom is our aged rat TPA model. Of course, TPA, the clot-busting drug, reduces the clots, but it also induces microhemorrhage. LG3, shown on the very bottom right part of this corner here slide, reduces that hemorrhage rate to 0 in this particular model. Again, restoring the homeostasis to the neurovascular unit, in this particular case, restoring the blood-brain barrier. And excitingly, in our most recent study, this is a pilot study in spinal cord injury, we see using a vasomotor motor score that if you give LG3, again, this 21 kilodalton protein that's cleaved off from an extracellular matrix protein, improvement in the animal's function as measured from day 1 to day 14 versus sham control. So this is a universal sort of finding we're finding. Frankly, we haven't studied an animal yet. We've studied other data here in diabetic mice and so forth where you see similar sorts of effects. Our plan forward here, we've secured IP across the second generation molecules. We have biologic exclusivity for our initial LG3 molecule, and this is going to give us composition of matter for the next 20 years or so. Our plan forward is actually very simple. We all recognize the difficulties of animal studies in showing effects. We've shown it across a range of different studies here. Our first goal is actually show proof of concept in humans. This is the next 2 years. First is a safety study in normal healthy volunteers to read out at the end of 2026. And then we're going to go into either a moderate to severe efficacy study or perhaps even a lesser study showing effects of this in patients with traumatic brain injury. TBI is advantageous for us because this very clearly reduces blood-brain barrier permeability, and through gadolinium, K-trans, DCE MRI, we think we're going to be able to pick that up. Not to mention the serum biomarkers, NFL, GFAP, and others that are acutely elevated in these patients, using these biomarkers, of course, with clinical outcome measures to establish initial evidence of efficacy. Now, I've shown some of these data, some of these things before about DCE. These DCE MRI correlates directly, really, with the initial, it's not so much the mechanism of action, but the initial findings of this particular drug that responds rapidly to LG3 treatment. And needless to say, there's a lot of different companies that have had great value added to these particular agents. This is some of our competitors. If we go ahead and look at the use of proceeds for our round, it's going to be almost exclusively in a $15 million raise toward the conduct of the clinical trials that I just outlined for you. We're very anxious to get into the clinic and show that this drug is working as effectively in the animal studies that we're going forward with. Again, the key number here at the end of Q4 of 27, we will have a readout on our TBI study using, again, the radiographic and serum biomarker measures along with clinical outcome data to show the effects of this really remarkable class of therapies that we are developing. And my time is getting short here, so I won't go into a great deal of detail. But of course, we'll have multiple options at the end of the readout of the TBI study. One of our great hopes is to have an exit that allows us then to use this particular drug toward chronic conditions. We have a second generation class of molecules that we think that will be amenable to lots of different diseases. Alzheimer's disease, of course, is one of the main ones, but also ALS, Parkinson's disease, and many others. We can use these assets and so forth in a way going forward to develop these as well for chronic conditions. And needless to say, this is a binary outcome measure for this particular company with extremely high valuation should we show that. So in summary, our team here, just shown here very quickly, we're working very carefully with experts in the field, Leonard Mookie here in the Bay Area, Ramon Aristide out of Penn, and Andrew Shih out of Seattle Children's Hospital are advising us as we go forward. There's our team on the bottom. In summary, Stream is developing a first-in-class, novel, pleiotropic, potent protein that restores homeostasis to the brain through the natural mechanisms induced by integrin receptors on multiple different cell types. This is supported by field-leading research. We're ready for the clinic. And we believe there's a very large value inflection point to be had following our initial studies in patients. Thank you very much.